Our next speaker is Joe Sexton. Joe is a NASA Earth Science Fellow and Doctor candidate in Landscape Ecology Lab at Duke University. He earned his BS at the University of Florida in Wildlife Ecology and Conservation and his MS here at Utah State in 2003. And what he's going to talk about is his work that he did here while he was at Logan, and this information is going to be published very shortly in Ecological Modeling. Title of Joe's talk is Habitone Analysis of Quaking Aspen in the Book Cliffs of Utah, Effects of Site Water Demands and Conifers on Aspen Cover.
Okay, so Habitone Analysis of Quaking Aspen in the Utah Book Cliffs, Effects of Site Water Demand and Conifer Cover. Dr. Campbell's talk was a very good segue to this, because this is coming from like the buzz hauling world at University of Florida, and then here ecosystem management is kind of one of the guiding principles in my master's research. And so the master's committee was larger. Fred Baker was also on it. Terry Sherrick was also on it. But in this, what I'm going to present here is this kind of a condensed version that Dale and Doug and I kind of put together for this publication. But as with all things, this came together with support from a lot of different organizations and a lot of different people. Most prominently, the Rocky Mountain Research Station of the Forest Service here in Logan, the Bureau of Land Management Vernal Field Office, and the Remote Sensing GIS Labs here at Utah State, and formerly the Landscape Ecology Modeling and Analysis Center. So we, to kind of zoom out and take a more macroscopic approach, Quaking Aspen is in decline over a very strangely speckly portion of the western United States. But we've got 50 to 95, that range, percent declines over large sections of its range. And when you have declines that are this widespread, the factors that are occurring in each individual place are going to vary. So we've already heard that the disease is widespread and very complex on its own. But then we've got climatic constraints and fire and succession to conifers and a large driver of ungulates. So what we need to do is kind of come up with a first approximation to determine not what to do to manage aspen, but where it can be done to greatest benefit. Like Andy Leidoff, I'm probably also, maybe not so much a toddler in Quaking Aspen, more of probably a wayward son, in that I'm living in North Carolina. But so this room is full of far greater experts on what to do with aspen when your feet are on the ground. But what kind of remote sensing and GIS can bring to the table is a more macroscopic approach and say, well, what are the broad scale constraints on aspen distribution? So what we're trying to do is go from this coarsely observed aspen decline. We know it's in decline, and we know we want it back. To go through kind of some sort of ecology to tell us, well, what is the potential of the land in every location, some kind of a map to get what kind of aspen can we expect if we do manage. And then bring that back and get that into a very digestible form of communication for the land managers to say, okay, we can take this and manage the land as we see fit in our district. So the ecology, that little fuzzy box on the top, is habitat modeling. But in this case, since we're dealing in very fine scales, we want kind of a sub-pixel analysis, and these are going to be Landsat-sized pixels, 30 meters on the side. We want to be able to see the kind of the percent cover. And so we're just going to generalize habitat modeling. You're used to habitat modeling, seeing a habitat map is kind of a black and white map. Either it can exist or it cannot exist. But if you look inside those pixels, and even in something as discrete as aspen, you've got edge pixels and you've got conifers coming up through, the pixels are not going to be pure. And so we need a way to kind of get a little bit closer, if not perfectly, but get closer to a percent cover estimate. So as kind of an annoying contraction or annoying augmentation of our jargon, a contraction of habitat and ecotone, we'll just call it Habitone. Try not to be annoying. So habitat, in the strictest sense, is the spatial projection of the realized Hutchinsonian niche, and I'll get a little bit more into that. And so Habitone is the spatial projection of a more continuous response. Think of it as a curve instead of a step function. Niche and habitat are binary, but response and Habitone are continuous. So think grayscale maps instead of these black and white maps. Just the same way that you wouldn't ask a hydrologist to model water flow on a binary DEM, high and low. We're trying to maybe model Aspen percent cover, or even the things that depend on Aspen cover, water flow, nutrient accumulation, things like that, on a continuously valued map of Aspen and Aspen habitat. And methodologically, niche and habitat, we use classification methods. But response and Habitone are actually simpler. We use regression methods, which classification methods imply or assume, and then go one step further to turn them into discrete measurements. So up until you get to the interpretation, which we're not used to, the methods are simpler. Okay, so consider the concept of niche. This is the foundation of habitat modeling in general. If you have and as Hutchinson put it, an n-dimensional hypervolume. Think of all the factors that lead to aspen presence or absence on the ground. This could be soil moisture, this could be solar radiation, this could be conifer density, another one could be ungulate density. All these different things kind of form this very high dimensional box, and somewhere in that box aspen can exist. Everywhere else it cannot. And so every point in this box will say this is aspen's positively valued habitat. Aspen can live in that portion of its environment, but not anywhere else. But, as you recently saw, if you're going to put land cover in boxes, the measurements that go into the habitat model, you're going to miss a lot of the real world variation. You'll get your pure aspen here, and you'll get your pure non-aspen here, but what about a very large chunk of the landscape? So what we need are more continuous methods to get a little finer resolution for monitoring, essentially. So you're going to have to kind of fuzz out that box of that estimate of the niche, and you still have think of the same ecological axes: water, light, soils, predation, all these things. But your box now, instead of having a subset of a box inside it, now has kind of a cloud of points inside it of percent cover. And the way that you estimate clouds statistically is with regression. And this is just a very simple cartoon. Think of this as any one, or in high dimensional space, kind of a PCA kind of view of the world, all of your environmental variables. And this is your measured cover of aspen. This is kind of a tradition in subpixel analysis. The membership of the pixel X in set aspen increases with whatever this variable we're calling. And the green line is the response, and 1 minus that green line we can think of as a constraint. And then the errors of every point on this will be a pixel out on the landscape, is the indeterminacy. And we can model not only the response and the constraint, but also the badness of fit of the model, which will tell us if these pixels are manageable. If aspen or the organism is tightly constrained by its environment and that fit is absolutely perfect, which usually occurs around zero cover, we don't need to bother managing it. So, application of the method. This is a map of aspen soil habitat, binary, over Utah and Colorado. And you can notice that here's the Uintas, and this is the Tamples Plateau. We are, I think, somewhere around there, maybe. This Tamples Plateau Book Cliffs area is really the only biogeographic link between the Uinta Mountain, or the Utah Rocky Mountains, and these larger Colorado Rocky Mountains. So if we're looking for things like biodiversity that are over long timescales, they're going to be migrating between these areas, this is kind of a pinch point in that ecological network. It's 10,450 hectares that look something like this. This is facing south out over, if you've been to Grand Junction and look north at those giant cliffs, if you were to jump over there, you'd fall down the cliffs. So we've got the vast majority of the landscape is non-forest, and it's Cercocarpus, Amelanchier, sagebrush, steeply dissected parallel canyons, Douglas-fir kind of sprinkled throughout, and then these giant aspen clones. And the landscape roughly divided between aspen and Douglas-fir once the non-forest is taken out. So, first thing we have to do is measure the aspen cover. And on any greater scale, this would be done with aerial photos, maybe by satellite imagery. But here, the topography was steep enough and the vegetation was low enough that we could walk out every one of these ridge lines in the area, take USGS quads, and just trace the clones, which are very discrete, onto the USGS quads, digitize those polygons, rasterize those polygons, and then coarsen them so that we could have 30 meter resolution cells of aspen cover. Now at this point, the interior of the polygons, which are really just a collection of adjacent pixels, are going to be pure aspen, and only the edges are going to have a cover somewhere between 0 and 100% or 0 and 1 fraction. So then, to get the Douglas-fir, we selected 2 canyons in the area randomly, and since it's so diffusely scattered, we can't really map every single location of Douglas-fir. And what we had to do was go to 139 plots in these canyons and take standard forest mensuration and apply the forest inventory analysis. What are they called? The canopy structural model to get some estimate of canopy cover. Those results were then used to train a regression of Landsat 7 thematic or enhanced thematic mapper multispectral variables. So it was a remote sensing regression to get percent cover of Douglas-fir. And so from the Quaking Aspen, we subtracted the Douglas-fir cover and got to decrease the interior cells from cover of 1 to somewhere between 0 and 1. So this is what the final map of aspen and Douglas-fir cover looked like. The non-forest is just transparent. You'll notice that we kind of have this north-south gradient. The Douglas-fir, this is a lower elevation to the north, and higher elevation to the south as you kind of come up to this rim. The Douglas-fir is more on the northern end and lower elevation, but seems to be creeping up the canyons and invading the aspen stands, mostly on the intersection of their 2 ranges, but also on these higher elevation aspen clones or stands. So the environment. And this is a greatly contracted version of the original, and so I won't go into the 96 other, or well, I guess 92 other predictor variables that we kind of just churn through, looking for the environment, the relevant environmental variables. But it really did in this area. The aspen was tightly constrained by the potential evapotranspiration, and so to get potential evapotranspiration for this area. It's essentially in a GIS, it's a hill shading model. You have a digital elevation model, and you can take a virtual sun and shine it on that and get the amount of direct beam solar radiation, shortwave solar radiation, on each pixel of that digital elevation model, and then use kind of a view shed analysis of the sky to get the diffuse radiation. So we have 2 components of the radiation environment, and then to get the heat, we use the Prism monthly mean temperature data. That's an average of 1970 to 1990, I believe. So this is what these maps look like. In March, the potential evapotranspiration is expectedly low, and in April, as the growing season begins, the landscape obviously lightens up and becomes more homogeneous as the sun angles are pointing more directly down on the landscape. You don't get as much shadowing by the topography. So in June and July, the landscape is very homogeneous, but then in August we're getting into the later season, and the landscape is really starting to differentiate more on potentially evapotranspiration. In October, it's darkening again, and then we didn't do this for the non-growing season months, basically because of the input data had large anomalies. So the main step in a habitat or a habitone analysis is an overlay, where you just take every pixel of, well, ideally you would take every pixel of your Aspen map and intersect it through your environmental variables to do a regression. That's computationally prohibited for a landscape of hundreds of thousands of pixels. Actually, probably not today, but then it was. And so we took a 1/10 sample and regressed the Quaking Aspen cover on all of these variables. Now, when you do a kind of a subpixel classification or a continuous analysis, you still have the variability and therefore the information in your data. So you can always degrade it to do kind of a more traditional binary habitat map. And so this would be aspen presence and absence over every month of the growing season as viewed on its PET. And so you'll notice that both the aspen and non-aspen cells track this overall seasonal PET pattern. But as the growing season, but the aspen patches are always slightly lower in potential evapotranspiration. And if you were to just do this statistically, you would not get significance. However, when you're looking at an area and you're looking specifically at that area, the statistical significance is not as important as just the fact that there is a pattern. And so so you notice, one, that they both track the overall pattern. Two, that aspen is always lower potential evapotranspiration than non-aspen. And then three, that the aspen-non-aspen difference in potential evapotranspiration diverges as the growing season progresses. And that's partially because in these June and July, there's really not that much variability to play with in the data. But they are still, despite that, they're still diverging. And so we can kind of attribute this to accumulated moisture stress throughout the growing season. And then if you kind of put that information back in your data and have each pixel still be the expected concentration, so not presence or absence, but the expected aspen concentration or cover in every pixel, you'll get this again following so March, April, May, June, potentially evapotranspiration. This is your expected aspen cover. You see that it follows that PET trend. Again, we've turned that previous graph kind of on its side and are looking at it in a different way. And the early growing season is tight. The difference between in PET of aspen and non-aspen cells, even in a continuous viewpoint, is not very large. But as the growing season progresses, that those areas tend to diverge, and that's just shown here again. Another thing that is interesting is that the expectation of aspen in pixels is always 0 at the high end of the growing season. So we definitely have a drought constraint. But we don't always have we actually never have a perfect topographic shade constraint. This is what we can interpret this low end of the PET range in any given month. So some months it's a little stronger than others, that we can see that there's that the topographic shading is having an effect, and there's somewhere in the middle of the PET range between topographic darkness, which never gets completely dark, but topographic shade and drought, we get the highest expected concentrations of aspen. And this is kind of a nice little glimpse. This September one is a nice little glimpse of a classic niche response curve. So this is all interesting kind of ecologically, but we also like to know how good our models fit. And one of the things with doing these raster analyses is embracing your error and actually interpreting it. So these are just two different estimates or ways to calculate the goodness of fit of a model. And you'll notice that early in the growing season and late in the growing season is better than that middle growing season because there's not very much variability to play with in either end of the growing season. In the middle of the growing season. So these are better than this. And so if we're going to just model it on any one month, which we don't really need to do, but it's really the ends of the growing season that are limiting Aspen in any one location. It's also interesting to note that the goodness of fit is a little bit higher on the autumn end of the growing season than the spring end of the growing season, which again says that it's not that darkness, or it's really accumulated growing season moisture deficits in the soil. So now this is kind of where it gets a little bit more intuitive for the managers and people that kind of work on the ground. I've just noticed that if you give somebody a map, they really kind of understand what you're talking about because they know their own land. And so what we can do is map this back out, and this would be identical to a habitat map if we took every pixel that wasn't yellow and turned it blue. But we can notice that there is different. The PET environment is constraining the cover of Aspen to varying degrees across the range of PET, and so every pixel with its unique combination of potentially evapotranspiration values can, in the model, support a unique combination or a unique concentration or cover of Aspen. And over this, I've overlaid the measured Aspen stands, and you can tell in this inset that it's a really good correlation. The correlation is 0.514 between the model and the expectation and the observation, which means that our system is fairly well constrained by PET, although there is a lot of room for management. Since we can't change PET appreciably, we have 1 minus 0.514 ability to manage this system. And again, this is actually kind of a limitation of the model in that it's very, very statistical, and there's not that much ecology other than the variables we put in that PET and Aspen cover. So the Aspen cover is constrained to equal the Aspen expectation. And so all those, you might have noticed that the Aspen cover in every pixel was constrained to be around, 0.35 at the highest end. And I really haven't figured out a way to adjust that in an ecologically informative way. So you can still treat it as ordinally very correct, but the clonal nature of the Aspen was not included in the model, and so that's a potential improvement. Another interesting thing to note is that the Aspen expectation and Douglas-fir cover are also fairly well correlated. It's not very strong, but that just says, and remember that the Douglas-fir was mostly up here and the Aspen was down here. And so in the past, these might have been Aspen, but now they're covered by Douglas-fir, which means the system has been progressing in this dynamic long enough to remove the Aspen. We can also, just like we mapped the expectation, we can map the deviance or the indeterminacy. And this is, so you'll notice that it looks very much like that Aspen map, and that's because the Aspen indeterminacy and the Aspen expectation, or the response, or the niche value, are correlated to 0.94, which tells you that when there is no Aspen, the environment tightly constrains it to not be there. But when there is Aspen, the environment doesn't guarantee, the PET environment definitely does not guarantee Aspen, but in many, many cases it can guarantee that you're not going to get Aspen. So again, Aspen indeterminacy and Aspen cover, that kind of points at that same result, 0.514 correlation. And Aspen indeterminacy and Douglas-fir cover, there's a decent, not great correlation, but that tells us that it kind of suggests that Douglas-fir is removing a lot of the Aspen from this area. Now, again, with the embracing error, we can take the observation, the aspen that we measured on the landscape, and subtract it from the aspen expectation. And this gives us a general idea of what we would expect the dynamics to be doing. Are we losing aspen in this area? Are we gaining aspen in this area, given the potential evapotranspiration environment? Now here's kind of the punchline of the whole thing. When we have all of these pieces of information, we can code this in a logical statement, and normally we just use classical Boolean logic to get at where the areas, the exact pixels that can be managed given this model and given what we know about management. But since all of the data in this case are continuously valued, we have to use fuzzy logic, and I'll kind of translate this fuzzy logical statement here. So just the heuristic model of aspen manageability is the absence of aspen, so we're looking for cells that don't have aspen, and aspen expectation, so absence of aspen and we expect aspen, and the proximity or the cover of aspen in the 8 cells adjoining the cell that we're looking at. And so we have to have aspen in close proximity because we haven't, from what I can tell, been very successful in planting aspen or using seedlings. It's just probably prohibitively expensive. And so this is what you come out with running the analysis all the way through, and you get kind of these areas. This probably has Douglas-fir in it. A lot of borderline areas around the aspen clones, and generally. Given this model, which is very heuristic, we can estimate in one management cycle, if this whole area were to be managed, we would expect 14.63 hectares increase, and that's a 2.46% increase over the current cover. So that kind of completes that circuit, that adaptive ecosystem management circuit of kind of monitoring a decline, taking it through the ecology, and then bringing it back to the land managers where they can do their job better than we can tell them how to do it. And then the cycle would start again with more monitoring. Which brings me back to a word from our sponsors.